Hi friends, this is Vishnu here and welcome to my channel Tips, Tricks and Concepts by Vishnu. Many students have told me that evolution chapter and some of its concepts like Hardy-Weinberg principles are very difficult for them. So in this video, we will deal with Hardy-Weinberg principle, the major concept of the chapter evolution. So to learn such concepts, such difficult concepts more easily with certain tips and tricks, please subscribe my channel and share it with your friends too. So now let's go to our topic. So Hardy-Weinberg principle. Before going into the concept, we have to learn certain words which are commonly used here. That is population, alleles, etc. So what is meant by a population? A population is a group of organisms of a species living in a particular area and are able to interbreed among themselves. So there are three conditions. There is organisms of a species, then living in a particular area and able to interbreed among themselves. So every individual in a population has certain characters and each of these characters are coded by certain genes in their chromosomes. And each of these genes are actually present in pairs, and each of these pairs is called as each one of that pair is called as the alleles. And these alleles of a particular gene or a locus, that is the position of the gene on the chromosome, are present on each homologous chromosomes. So let's see what their importance is in the Hardy-Weinberg principle. We can find out the frequency of occurrence of the alleles of a gene or a locus in a particular population very easily. And Hardy-Weinberg principle states that at a particular conditions, at certain conditions, the frequency of occurrence of these alleles of a particular gene in a population remains constant over generations, and in such populations evolution is said to not occur. That is, in your text it has been denoted that allelic frequencies are always stable and is constant from generation to generation. Let's make it clear by using an example. Here we have three genes, that is three alleles of the same gene coding for the character eye color. That is, one allele is capital B coding for black color of the eye, and the other is small b which is coding for the brown color, or you can take it as blue color. So if in a population, say like this population, if we have 100 people and out of these some people are with black color eye and the others with brown color eye, then the frequency of the occurrence of alleles for brown color and black color will be constant over generations under certain conditions. Also remember that this is a population of 100 people, okay, 100 human beings alone, and if the eye color is taken as the gene or character, then how many alleles of eye color will be there in this population? We have two alleles for a particular character, two alleles for this character too, that is capital B and small b. So 2 into 100, 200 alleles. Let's take, for example, in this population with 200 alleles, we have 120 alleles as capital B, that is with black color eye, and 80 alleles coding for brown color. Okay, then that 120:80 ratio, that is 3:2 ratio, remains the same over generations. Okay, that is the same ratio will exist for the next generation of the same population. Okay, this is what is meant by Hardy-Weinberg principle, and there are certain conditions which I will mention at last. Now let's take the frequency of occurrence of allele capital B, that is for black color, as P, and for allele small b as Q. Then now we have to find what will be the genotypes here for this eye color. We have capital B, that is homozygote for this black color eye, dominant trait, and small b, that is homozygote for the recessive trait, brown color eye, and we have heterozygotes capital B, small b, and there will be two such combinations and hence two capital B, small b. So now what will be the frequency of occurrence of this capital B people in this population? P into P, that is P square. That is, we have two alleles and both that alleles should be capital B, and so the probability of one allele to be capital B is P and the other allele to be capital B is also P. So we have P into P. Say for example, if we toss a coin, what is the probability of getting the head? 1 by 2. And again, both, if we toss two coins at the same time, what will be the probability of both being heads? 1 by 2 into 1 by 2. A little max here. That is, the same is happening here. That is P into P, P square. Similarly, for the frequency of occurrence of small b in the population is Q square. And the other one, that is capital B, small b, will be 2PQ. So, what will be the total formula? P square plus 2PQ plus Q square equal to 1. Okay, that's otherwise P plus Q the whole square equal to 1 or P plus Q equal to 1, which favors our initial result of Hardy-Weinberg principle. That is, the sum of allelic frequency is always a constant. There in our example, it was 3:2. So, the proportion of B in the population, capital B alone in the population is 3 by 3 plus 2. That is 3 by 5 and small b 2 by 5. So, 3 by 5 plus 2 by 5, 5 by 5, that is 1. And that is proved here. That is P plus Q equal to 1. And where do we use these equations? If we are asked the allele frequencies, we can tell the equation P plus Q equal to 1. If we have to find the genotype frequency, we have to use the equation P square plus 2PQ plus Q square equal to 1. So, for your exams, you may be asked to find out the frequency of heterozygotes or homozygotes of dominant trait, homozygotes for recessive trait, and so on. So, here let's look at how many heterozygotes, or what will be the probability of heterozygotes in the population. That is 2PQ. That is 2 into 3 by 5 into 2 by 5. That is 0.48. So, now the mathematical representation of our Hardy-Weinberg principle is over. Now, let's go to the factors affecting this Hardy-Weinberg equilibrium. Remember that Hardy-Weinberg equilibrium or genetic equilibrium is nothing but what is said about the frequency of alleles. That is, the frequency of occurrence of alleles in a particular population remains constant over generations. So, now let's see the factors. The factors are gene migration or gene flow, genetic drift, mutations, natural selection, and genetic recombinations. Also, we can add another factor, that is non-random mating of individuals. So, now what is gene migration? As the name indicates, the people with a definite genotype and definite set of alleles migrate from a particular population and enter into a new population. So, new genes are added to the new population. At the same time, some genes are lost from our old population. So, let's see an example. Here we have a population A and a population B. So, a set of people P goes from this population A to that population B. So, there will be more people or more genes in that population B, and that set of P people will be removed from or will be lost from the population A. And in this way, some exchange of genes have taken place. Now, if such migrations, gene migrations occur in multiple times, then it will be called as gene flow. Now, the second factor affecting this genetic equilibrium is genetic drift. As the name indicates, it is similar to our gene migration, but only difference is that here it occurs by some chance events or by some unprecedented events. For example, some natural calamities or some forest fires and some excessive hunting, etc. Here, two effects are associated with genetic drift, that is founder effect and bottleneck effect. And for these two effects, please look at my next video where I have explained in detail, that is with examples. Now, I will just mention about founder effect, that is, if the change in the allele frequency during the genetic drift was so high that it becomes a new or a different species in that place, then that effect is called as a founder effect. That is, if a group of organisms drifted to a place where there were no such species, then it will be new to that land, right? So it will produce its own population in that area. And so such effect is called as a founder effect. Also, that originally drifted population is called as a founders because they are the founders of the new population in the new area. And if you still have doubt, please refer my next video. So now let's go to the next point or the next factor, that is mutations. Mutations are nothing but sudden variations in the genome of an individual or a population. And it may be due to many factors. As you know, there are many agents for these mutations, physical agents like UV rays, chemical agents, and so on. And if due to some agents the mutation had occurred in a particular chromosome, in a particular gene, then that agent itself or that mutation itself introduces a change in the character coded by that gene, and hence mutation are an important part of evolution and also affects the genetic equilibrium. Now the fourth factor, that is natural selection. Now what is natural selection? Natural selection is nothing but a process in which heritable variations allowing better survival are made or are enabled to reproduce and leave more progeny. This means that if an organism has certain inherited variations and it enabled it to survive that condition, that is adapt to that conditions, then the nature favors that organism and that inherited variation, and hence that organism can reproduce and leave more progeny, and hence that population with such variations are favored, and this is called as natural selection. And natural selection can operate in three various ways, that is as given in the figure here. So here the three types of selections, natural selections, are the stabilizing type, then directional type, and disruptive type. So let's understand each of these with suitable examples. For that, let's invite 4 new friends, that is 4 different mice. That is a white colored mouse, then a cream colored mouse, a brown mouse, and a dark colored mouse. Okay, so let's assume that they live on a certain land, and for example, if the land is soil, is mostly soil, and it resembles the color of the yellowish or the cream colored mouse and the brown mouse, so naturally it will be safe from the predators. And the white and the black colored mouse, that is the extreme ends, will be easily seen by the predators and will be caught. So the population of the white and the dark colored mouse will be very small, while the cream colored and the brown colored mouse will be very favored by their color, and hence the nature favors it too, and they can easily reproduce and produce more progeny, and so its population will be in the peak. So here in stabilizing selection, the middlemost characters or the medium characters are, or medium phenotypes are favored, while what happens for the extreme ends, that is the extreme phenotypes, that is white and the darker colors, are actually not favored by natural selection. So this is the stabilizing set of selection. Now the next one is the directional type of selection. Now here let's assume that the land is a rocky area, so a darker background. So naturally which will be more preferred or more favored by nature? The darker mice, because they cannot be seen easily by the predators, and then followed by brown, then the cream, and finally white. So the darker mouse will produce more progeny, and hence it will be the higher population at the higher population end. Now here it favors the extreme phenotype, and hence it is a directional type of natural selection. That is, the nature favors to a particular direction, that is to a particular extreme. And similarly, if the background was a relatively white background or a relatively bright background, then naturally the white mice will be saved and it will produce more progeny. Okay, so the only difference here will be that the peak will be towards the opposite extreme. So it is directed towards the opposite extreme, and it will be also a directional type of natural selection. Now let's go to the next type, that is the disruptive type of natural selection. If the land supports both the extreme ends of mice, that is both the extreme colors, the white and the darker ones, then they both can produce more progeny and hence a big population, that is two peaks at the extreme, while the middle phenotypes are less favored. So a peak which was actually to be formed is actually disrupted. Okay, a single peak has to be formed here as in stabilizing and in directional selection, but that was disrupted due to these two peaks. So it is called as the disruptive type of natural selection. Okay, now let's understand some real life examples. Okay, first of all, for stabilizing selection, we have the phenotype as the human baby weight, okay, at the time of birth. So the birth weight which is usually favored for better survival is nothing but between 2.5 to 3.5 or just 2 to 4 kilogram. So the peak population or peak survival rate will be in that weight group, okay, that birth weight from 2 to 4 kilogram, and below 2 and greater than 4, less chance for survival. Okay, now let's see an example for the directional type of natural selection, that is industrial melanism. In 1850s in England, that is in the pre-industrial period, the trees were all covered by white colored lichens. So the white-winged moths living on these trees were not easily seen by the predators, while the dark-winged moths in England were easily seen by the predators, and hence the population of white-winged moths were very high and dark-winged moths very less. But what happened after the industrialization? That is, in about 1920s, the trees became covered with soot, dust, etc. And now the predators could see the white-winged moths more easily than the dark-winged moths. So gradually the white-winged moths numbers came down while the dark-winged moths increased in population. So here the natural selection in the pre-industrialization period favored the white-winged moths, and hence the peak population was towards that direction, that extreme end. And in the after industrialization, post-industrialization period, dark-winged moths were more preferred, and hence the peak population was towards the opposite direction. And these are good examples for directional natural selection. Now let's see an example for disruptive type of natural selection, that is London's peppered moths. In London, in rural areas, the population of the light-winged or the white-winged moths was higher, but in urban areas, the dark-winged moths were higher. This is because in urban areas, the dark-winged moths were able to hide themselves from the predators among the polluted surroundings. But in the rural areas, the light-winged moths were able to hide from the predators because the trees were all covered with white-colored lichens, as I have told in our industrial melanism example, and hence light-winged moths were able to survive there, while dark-winged moths not. But what was the case for medium-colored winged moths? They could not be seen in high population numbers, or they were found in very low numbers in both the locations. Why? Because they cannot camouflage themselves from the surroundings in both locations. So here the natural selection favored the extreme ends, that is the light-winged and the dark-winged moths. So here there are two peaks, and hence it is a kind for disruptive type of natural selection. So in stabilizing type of natural selection, most of the individuals acquired mean character values, but in directional type of natural selection, most of the individuals acquired character values other than the mean character values, that is towards one or the other extremes. While in disruptive type of natural selection, most of the individuals, that is more individuals, favored the peripheral character values, that is double peaks at the two ends, at the both ends of the distribution curve. So the natural selection is over. Now let's come to the next factor, that is genetic recombination. As you know, in meiosis and all, crossing over of genetic material takes place. So that crossing over is an example or is a method of genetic recombination, where the genes are exchanged between the chromosomes. So the genetic recombination produces a change in the character coded by the particular allele of the gene. So these are the factors that affected the Hardy-Weinberg equilibrium or genetic equilibrium, and they cause evolution. So now what are the conditions where this Hardy-Weinberg equilibrium exists? Let's see the conditions. The conditions are the population should be of large size, okay, and then there should be random mating among individuals of the population. Then third one, no mutations. Then no genetic recombination. No exchange of genes, that is no gene migration, gene flow, etc. and no genetic drift. So you can see that the factors which affect the genetic equilibrium should not be there. And in addition, the population size is very large. Okay. Also, in non-random type of mating of individuals, that is the preferential type of mating of individuals which usually exists among the organisms, this Hardy-Weinberg equilibrium does not exist. So this is all about Hardy-Weinberg principle. We have discussed about the Hardy-Weinberg principle, its mathematical representation, that is p plus q is equal to 1 and p square plus 2 pq plus q square is equal to 1, that is for genotype frequencies. And then we discussed the factors, and finally we discussed the conditions where the Hardy-Weinberg equilibrium exists. I hope you have understood my class, and if you have any doubts, please feel free to ask, and please watch the next video, that is genetic drift. And please don't forget to subscribe my channel, and bye bye for now.